is huge work and large organization funded by US government, European Space Agency, and various universities. Several collaborators listed here, and all have worked together equally in this project. Here is Dr. Bafin Murasani from Romania, Rupi Darsalani from US, Paula Majari from US, Jose Vicente Lafrente from Spain, Anka Bujoyanu from Romania. This is our colleague Ryan Tian from Faith with US. He has the US patent on nanowired delivery that we are using. This is Dr. Huan from Beijing. This is Professor Fung from CGS Juan, Dr. Chen from Beijing, Igor from Vladivostok, Manjulo from Vladivostok, Lars Wiglund from Uppsala University, and Halai Parnashal. First of all, I would like to mention that whatever I am speaking about this is our conclusion, and no governments are involved, and it is all responsibility goes to me. We are working on Department of Defense research project that says that military personnel are highly vulnerable to traumatic brain injury, which is quite obvious, and therefore whatever is available at the moment for their treatment is not sufficient enough. So therefore we try to understand how drugs can be delivered and how they can be more effective, because there is blood-brain barrier that stops the penetration of drugs in normal conditions and also under injury conditions where there is swelling and damage. So that's why we do research that all kinds of neuronal diseases are basically because of breakdown of the blood-brain barrier. In our central nervous system, there are several barriers. Blood-brain barrier is one. Blood cerebrospinal fluid barrier is another type. And Alzheimer disease is most common that we know, even layman knows that it is very common with the old age disease. But when you are working in military profession, either at vigilance, combat, or any kind of operation, there is lots of stress. And if you get traumatic brain injury, then you are more vulnerable to Alzheimer disease and even Parkinson's. As you can see here, that this brain appears to be normal, the ventricles are normal, but when we have mild cognitive impairment, the ventricles are enlarged, and in Alzheimer disease, they are much more enlarged at the expense of neural tissues. So that is the problems of Alzheimer disease. A cartoon showing also the same thing, that the ventricles are enlarged here, mild Alzheimer disease and severe Alzheimer disease. And so far, there are no any specific treatment that can reduce the events. This is just an example that military risk factors for Alzheimer dementia and neurodegenerative diseases. And traumatic brain injury is one of the important factors in developing Alzheimer disease in military personnel. Many authors try to understand the fingerprinting of amyloid beta peptide, and it is highly controversial because there are many people saying that amyloid beta peptide may not be the real cause of Alzheimer's disease, but amyloid beta peptides are neurotoxic, and that is one of the important aspects. People are trying to reduce the deposition of amyloid beta peptide in the brain. Here is an example of the normal brain after brain injury, and also the E level showing post-traumatic stress disorder, how the amyloid beta peptide can influence the whole brain structure. The same thing is showing here, but the important thing is seeing it is a combination of traumatic brain injury and also post-traumatic stress disorder. They are the worst cases. There are various regions in the brain, and they are not equally affected by amyloid beta peptide deposition. For example, in this case, this is frontal cingulate cortex, parietal cortex, occipital cortex. You can see normal PTSD, means post-traumatic stress disorder, traumatic brain injury, the third one, and both combination of traumatic brain injury and post-traumatic stress disorder. The spread of amyloid varies in this is also temporal. One of the earliest studies found that the factors responsible for Alzheimer's disease lies in plasma. And this is one of the examples here. Plasma factors contribute to Alzheimer's disease, published in Proceedings of National Academy of Sciences, one of the prestigious journals in 2018. This is one of the examples published in this country, Netherlands, Maastricht, that in Alzheimer's disease, there is a leaky blood vessels in the brain. That is one of the important aspects of developing Alzheimer's disease. Using animal experiments, this is the first report from our laboratory that there is a leakage of albumin in Alzheimer's disease model in controls, means animals having Alzheimer's disease. And here is, we have given a drug called cerebrolysin that is able to reduce the leakage of blood-brain barrier. So showing that leakage of blood-brain barrier is an important component of developing Alzheimer's disease. If amyloid beta peptide is responsible or it is coincidental, one can measure and find how it changes. This is an example that in cerebral cortex, amyloid beta peptide is very high. When we genetically infused Alzheimer's disease mouse model was developed. When we treated cerebrolysin, it is reduced. When we have given nanowired cerebrolysin, it is further reduced. This is the importance that how nano delivery of drugs can really affect the Alzheimer's disease process. Another important aspect is phosphorylated tau that is also increased in Parkinson's disease, Alzheimer's disease, and even traumatic brain injury, sleep deprivation. So we measured phosphorylated tau, and here is in Alzheimer's, it is high. Cerebrolysin treatment has reduced, but when we give a nanowire cerebrolysin, it is significantly reduced. Opening the point that nano delivery of drugs are important in developing or reducing neurodegenerative diseases. The next point is that whether developing Alzheimer's disease is exacerbated in traumatic brain injury. We have a model of traumatic brain injury in which I'll show you. This is the model of traumatic brain injury. A rate of 114.6 is dropped over the skull, and it produces 0.224 Newton impact. That is quite similar to mild traumatic brain injury that is occurring quite normally across the world. Morphological examinations show that although the right side is injured, but the left side, using counter-coup mechanism, very well known to neurosurgeons across the world, is much more damaging than the direct injured part of the brain. And old data showing how traumatic brain injury is prevalent in US military personnel. The network is affected by traumatic brain injury. As you can see here, working memory is lost, motor planning is lost. We advocate through scientific technology that nano delivery of drugs required to treat this neurodegenerative disease. These are titanium nanowire we use, and my colleague Dr. Ryan Tian has a US patent on this. So therefore, they are developing the drugs tagging the nanowires, and we are using this. Dr. Ryan Tian, and this is Dr. Wang. They have the US patent of nanowired technology. You can also nanowire stem cells. And because when you nanowire stem cells and inject in the body, they live longer and they have better influence than normal stem cells. Well, this is great slide, but anyway, I want to show you that when we have given amyloid beta peptide infusion together with traumatic brain injury, the blood-brain barrier, brain edema, neural injury, amyloid beta deposit, and number of albumin cells were significantly increased. This is an example. We also use the combination because this is very important. Not only drugs, but they are potentiated by delivery of nanowired stem cells. This is an example showing that untreated group, nanowired mesenchymal stem cells alone, nanowired cerebrolysin, and combination of nanowired cerebrolysin plus mesenchymal cells. This is the significant neuroprotection in Alzheimer disease. Another enzyme is important in brain injury and Alzheimer disease is the neprilysin that is reduced because this is the amyloid beta degrading enzyme. And this is an example that amyloid beta, they are reduced, and also in GFAP level, they can be found. This is another table that's showing that when we have given nanowired cerebrolysin plus nanowired neprilysin in Alzheimer disease, the changes are significantly reduced. This is an example of hippocampus, and you can see that in the middle, traumatic brain injury plus Alzheimer disease, cerebrolysin treated, they are better than the untreated group. But when we have added nanowired neprilysin, the cells are definitely better than the second. The real reason is stress that is caused by military deployment, either they are in combat operation or for vigilance or even peacekeeping missions. And the stress is very important. They are confined to single posture in various ways, and therefore we examine the effect of restraint because sometimes they are very fixed in one position with gun and many other things. So they are just like immobilized. So we have used restraint stress 4 hours for 1 week and then given the Alzheimer disease. This is amyloid beta peptide in untreated, and here we have given nanowired cerebrolysin plus monoclonal antibody to amyloid beta peptide and serotonin receptor antagonist. You can see that they are significantly. These are the neuronal cells, and similarly they are significantly decreased with these treatments. This is EGA1 because cells and also microglial cells are affected, and this is the example in which you can see that the combined treatment has reduced the microglial regression measured by EGA1. CD86 is also increased significantly, and they are also decreased by these combination effects. At electron microscopy, as you can see here, that the neuropil is much better protected when we given the combined treatment. Therefore, the combination therapy appears to be quite important as compared to each individual given alone. Questions are asked why nanodelivery of compounds are important. it means that if you nanowire delivery use any compound that will act? The answer is no. Here, this is an example. There are 4 compartments, and here is spinal cord injury. The only one compound, I cannot mention the name for other reason, SCI01, is the best, whereas the other nanowire delivery compounds are worse, and some of them are in the middle. So therefore, we have to choose the good compound that can deliver better result when we give using nanowired. One of the reasons how they are working, because there is a blood-brain barrier, they penetrate the endothelial cells and release their compounds inside the brain. Therefore, they are active for long period, and this could be one of the reasons why they are more effective than the conventional drug delivery. And leveling of antibodies was recognized by National Innovation of the United States. They have given us this award. We are publishing some books and volumes to advocate that nanowire delivery or nanodrug delivery is key to reduce neurodegenerative diseases. This is one in progress in brain research. This is the latest book release. I have a copy here. You can see that if you like, by Springer. Then there are also International Review of Neurobiology, progress in brain research. And they behave very similarly even in cancer cases. Therefore, this is summarized in another book. And these are 2 volumes are coming, International Review of Neurobiology 171 and 172. So we can see that combination of drug therapy or antibodies or stem cells together using nanodelivery are much more efficient than the conventional drug delivery. Nanomedicine appears to be future for the treatment of these neurodegenerative diseases. This is a large project supported by various organizations. These are my co-authors. I showed you earlier. Various support from different organizations. My collaborators. I am from Uppsala University, and I think you very much for taking part.
Thank you, Professor.